So Watson and Crick were able to finally deduce the correct structure of the DNA double helix. While that would have been a major, a major finding for, biology, for science in general, their contribution to DNA sciences, to biology, to biochemistry and chemistry went even further than that. Part of what also helped seal the deal in them winning the Nobel Prize was their contribution to determining how the DNA double helix acts, okay? How it is replicated, okay? Because you got to remember, at this time there are scientists cell biologists who are, furthering the research into cell cycle. We're studying S phase of interphase, and they are sitting there and seeing these chromosomes replicate and how the chromosome numbers would be doubling. And they couldn't figure out how this worked. They now knew the DNA double helix. They knew those, the proteins were there for structure, not for information. Watson and Crick also examined this question. The question that other labs are also trying to answer, trying to look at when it came to DNA replication is, how does it work? Okay. We knew we have a double helix composed of two anti-parallel strands. Remember, we talked earlier chapter, the anti-parallel means that as one strand's pointing up, the other strand's pointing down. This orientation allows for their nitrogenous bases and the nucleotides to interact. But the question was, how does this allow for duplication of DNA? How does this allow for new chromosomes to be duplicated and done so that fidelity can be maintained? Here, fidelity corresponds to, wherever there's an A, the new copy will also have an A. Wherever there's a T, the new copy will also have a T. Same for the G, same for the C's. So that the copy is nucleotide for nucleotide exact replicate, an exact replicate of the original. Start off with the parent molecule. Copy is made. You now have two daughter strands. And how can those daughter strands be made that nucleotide for nucleotide, they're the same as the original? Excuse me. What Watson and Crick were able to figure out was the original double helix would be split apart. The two anti-parallel strands would be pulled apart and would act as templates to lay down new. So the two new, or quote unquote new daughter strands would actually each have one strand from the original. So that way, when the original is separated, wherever there's an A, a new T would be added. Wherever there's a G on the new strand, or a C, a G would be added. Where there's a T, an A would be added, and so on. And the opposite. would also be true. Wherever there's a T, an A. Wherever there's the G, a C. So when you look at the two strands, ACTAG, ACTAG, the original TGATC, the new TGATC. So the opening of the original parent and using that as template was a revolutionary thought, because many others labs were trying to disprove other competing models. Watson and Crick called this the semi-conservative model. Okay, you're somewhat, hence the semi, conserving the original. The daughter double helixes were based on the original, and the original was somewhat conserved in the daughters. Other competing models looked at total conservative. Somehow the double helix, the original parent, would be red, and copies made from that. And the copies would then be a template for making more copies. Or a dispersive model. Its portions would be red, portions would be old, portions would be new. And, well, there's problems inherent in this. What would happen if there was a mistake right here? Okay, what should have been an A now is a C. Well, it turns out all of the copies from here on out will have that mistake. Half of all resulting double strands would have a mutation. Half would still be like the original. That wouldn't work. Logically, that wouldn't work, because that means that the ripples, the downstream ripples, what if you had another mutation, in the 10th replication? Well, that means that from the 11th replicant on, some of them are going to have the original mutation, they're going to have the second mutation, then there could be a third and a fourth, and no two cells in a tissue, no two cells, in the body could be the exact copies. And this doesn't work. This just does not hold out. So this cannot work in reality. The dispersive model, where parts of the daughter double helixes has part original, parts are new, this can't work either, because where's the machinery that would able to link together complete chromosomes? If only parts are being copied, parts here, parts there, and then linking it with original, blah, blah, too much error. You could end up with chromosome 1 missing huge chunks, chromosome 4 having too many parts, and that just won't work. So logically, that can't be it. We now know, by studying different cells as they replicate and studying the machinery involved in DNA replication, that the only model that could work is Watson and Crick's semi-conservative. The original double helix is split apart, and each of the strands becomes the template for a new strand. Fidelity is maintained.